Hello and welcome to this video on transpiration and the pedometer. In the video, we're going to look at a recap of transpiration, that's water movement through a plant, but also go on to speak about the pedometer, go into detail about how to measure the rate of transpiration using a pedometer, and also suggesting some example investigations where you would need a pedometer in order to complete it. To start with, transpiration in its basic form is the loss of water in a plant. Okay, so the loss of water, water is absorbed by root hairs in the soil and it travels up the xylem, that's the water movement in the xylem, up the stem, into the leaf, and is lost out of the leaf. Now they're lost through stomata. Stomata, depending on what plant it is and what type of plant it is, will have stomata on the underside or even a few on the top of the plant. This is sort of the ins and outs of water vapor and carbon dioxide and oxygen in photosynthesis and in respiration. That process of water loss as water vapor through the leaf is called transpiration. Now, the stages to transpiration, as our recap, I always like to start from the leaf. So water vapor is lost from the leaf through the stomata because of maybe a high temperature or it's something that's very windy and we have a lower water potential outside the leaf compared to inside the leaf. So obviously that water vapor is going to want to move out down its concentration gradient and leave the plant. Because it leaves the plant and it leaves the leaf, it produces another concentration gradient between the leaf and the roots. So water is going to want to move from the roots to replace that water that's been lost in the leaf. So water moves from the roots up the stem into the leaf to replace it. What we create a second time is another concentration gradient between the roots and the soil this time. So there is less water in the roots because that water's moved up into the leaf, so water has to replace it. And water comes from the soil, moves into the plant, moves into the roots, through the root hair cells, down its own concentration gradient, where there's more water in the soil than there is in the root. And this process, this transpiration stream, it's called, happens all the time. If you've ever noticed water loss in plants, maybe the grass on a summer morning, and you wonder, where has all this water come from? It was a dry evening, there was no rain. Why is the grass wet? Well, it's the dew. We always say it's the dew, but we don't realize that actually all that is water moving through grass. And because there's such a high pressure of water and water still wants to move and leave the plant, actually it's forcing its way through the stomata in the grass. And what you see in the morning is just a collection of water that's almost hanging, dripping from the stomata. But why is this important? Okay, we can have a look at transpiration and we can measure the rate of water loss through the leaves. But there's got to be a better way of finding it out. We can't just measure the amount of water in the soil. It's unrealistic. We can't collect water vapor above the leaves in like a bag, or I don't know what you'd use, put a pane of glass above it and just watch the water vapor condense. That's really unrealistic as well. Maybe you could guess. I don't know, I'll probably put a mug of water in your plant that's on the windowsill every day. It's about a mug, it's about 500 ml a day. It's not really scientific, is it? So biologists use a piece of apparatus called a pedometer. And pedometers can look very, very different, but they all do exactly the same job, which we will describe. So here are a selection of pedometers. They all look very, very different. The one that we have in school mainly is this one that you fix upright, but the other common ones are the horizontal lying ones. And a pedometer is just a piece of apparatus used to measure the rate of transpiration. And the way that happens is we have a chute that's been cut, and that's placed lots of Vaseline on there to seal it so we don't get any air leaks. and the apparatus is just full of water into a reservoir. There's two reservoirs really. There's a reservoir at the top here which helps you control the air bubble, and there's a reservoir at the bottom which replaces the water as it's used. So linking our transpiration to a pedometer, we know water is lost from the leaf. That water needs to be replaced because we've got a concentration gradient, so that enters the stem that's being cut. That has to be replaced, so it moves along from this reservoir that we've got in the bottom. That's why we need that reservoir there. And as water moves, this air bubble moves with it. So it's fair to say that the speed in which that air bubble moves is the speed in which the water moves. Now, this reservoir that you can see that's on the top of the pedometer, that reservoir there is to control and sort of orientate where this air bubble is. If you want to do multiple readings, you'll want to push the air bubble back and let it move, push the air bubble back and let it move. So this reservoir at the top, that's what it's used for. The faster the uptake of water from the reservoir, the faster the bubble moves. And that's what you're using a stopwatch or some kind of timer to time, and you're going to measure this distance. It's going to be a meter ruler or some kind of ruler with millimeters on. You're going to measure how far that bubble moves and how long it takes, more importantly. But setting up a pedometer is not easy. There are a few things that we have to follow all the time. And if your pedometer doesn't work or the air bubble doesn't move, well, there's going to be a number of reasons why. The first reason, it might be that you haven't cut the stem and set the entire apparatus. The whole thing needs to be set up underwater. Okay, so that's why we have those big sinks and sites. It's all set up underwater. You need to have a sufficient amount of water in the reservoir to place the air bubble at the correct spot. And getting that air bubble inside that tube is pretty difficult because it's been set up underwater. The last one is recording the distance that it travels over a specific amount of time. You could time how long it takes to move a centimeter, or you could just time how long it takes, how far it takes to move in a minute. Okay, you can pick what you want to do. It will all give you a rate of transpiration in the end. This one here, this cut the stem and set up underwater, that is the biggest sort of error that can occur. And even if you're trying to set it up and you do set it up underwater, it still might not work. Okay, if we have any leakage, so maybe the stem here hasn't got enough Vaseline or plasticine or whatever you're using to seal it, then the xylem will be broken. There'll be an air bubble in the xylem. As soon as there's an air bubble in the xylem here, then you will not get any water moving. Okay, the transpiration stream will be broken. But why? Okay, why use a pedometer? This pedometer mainly is used to sort of investigate the effect of change in the environment or even change in the plant. So different plants have a different rate of transpiration depending on where they have to grow, what conditions they grow in, what conditions they can survive in as well, what the humidity is, what the temperature is. So initially, it's used to prove that stomata exist mainly on the underside of the leaf by putting Vaseline on. So you put some plants in that have no Vaseline, and you gradually increase the amount of Vaseline that you put on. You might see a difference in the rate of transpiration, the speed at which the air bubble moves. You might also have changes to the environment, so changes to humidity, changes to temperature. You might sort of artificially control these conditions inside a laboratory, inside a school lab, so you can test transpiration. The more humid it is, the smaller the concentration gradient between the leaf and the outside air, so you have a decrease in transpiration. And overall, you get this graph by the end of it. You've got the distance moved by the bubble. In this case, it's by millimeters, but it could be centimeters, could be whatever. And the time as well, it could be in seconds, it could be in minutes, whatever you're recording. And here we've got a number of points plotted. You have to plot all the points along and it creates this lovely straight line. So the rate of transpiration here has been very consistent. Okay. If we have the bubble moving a further distance in a shorter amount of time, then we have a faster rate of transpiration. And on the opposite side, if we have the distance the bubble moved is shorter and it takes the same amount of time, we've got a slower rate of transpiration. So this gradient, this line, will tell you the transpiration rate. The purple line here is a very slow rate of transpiration. The red is getting faster, and the green is the fastest line. But why is this important? Why do we need a graph? Well, we need a graph to be able to work out the rate as a number. Okay, so to start with, it might just be a qualitative study that, yeah, the bubble moves faster, so it's a faster rate. But if you want some numbers to it, a quantitative result, then you will need to take the distance and divide it by the time. So in this case, we've got millimeters for the distance and we've got minutes for the time. So if I want to find the rate of transpiration for this particular experiment, I will pick some time. I'll pick a distance. We'll pick a distance first, which I've gone for 2 millimeters. This graph really should have numbers going up the axes, but miss them off. 2 millimeters, and I'll pick a time of 5 minutes. I've made it easy for myself. Okay, I'm not picking something down here where I have to sort of guess. Oh, it's about 1.2 millimeters. I've picked 2 easy points where they both intersect nice integers, nice whole numbers. And to work out my rate of transpiration, I'm taking what I said before, I'm taking the distance and dividing it by the time. So my distance is 2, my time is 5. So 2 divided by 5 is 0.4 millimeters per minute as my rate of transpiration. Okay, 0.4 millimeters per minute. There are a number of things that can affect transpiration, and this is the more higher level stuff, not just stating it, but also explaining it. So explaining why increasing light intensity will increase the rate of transpiration, why increasing temperature will increase the rate, why increasing air movement, that's how windy it is, and decreasing humidity. Why is humidity opposite to light intensity, temperature, and air movement? Explaining why they affect the rate of transpiration will get you those higher marks and those higher level questions. What you can go away and do is look at these 3 tasks. You can start by explaining how a pedometer can be used to measure the rate of transpiration. Now, that can be a method, and you can go and research particular methods, sort of pick one that's nicely worded, because it comes up quite a lot. Secondly, you can look at these factors that are on the screen at the moment and how they will increase the rate of transpiration. So all of these will increase the rate, but you've got to explain why. You've got to link it to concentration gradients, water movements, osmosis, all that kind of stuff. And also you can link it to work that you've probably done on hydrophytes and xerophytes and adaptations to minimize water loss from the leaf. Okay, that's number of stomata, that's the structure of the leaf to protect it from air movement, all that kind of stuff. So those 3 tasks I'd like you to have a go at. Hopefully from this we've got that, if I go back to the start, we've looked at water movement through a plant, we've explained what a pedometer can be used for, and also the pitfalls of setting it up, what can go wrong, what can cause the pedometer not to work. And also we've looked at investigations to require that will require pedometers and how to work out this numerical value of the rate of transpiration.